produced by the Howard Hughes Medical Institute.
In this video then, we'll look at the beginning of development, looking into the ovary and an unfertilized egg. It then gets fertilized by sperm, which travel down the fallopian tube. So here, millions of sperm coming along. Several of them will hit the egg and try to penetrate it, but one will win, as it were, go into the nucleus, and then there's a reprogramming process where the male and female nuclei have their genes set aside to be turned on and off for early development. Here you see early cleavage stages occurring, and this is one of the early growth phases. As the embryo moves down the fallopian tube, it's going to form an important stage called the blastocyst here in a few seconds. Of course, in real life that takes days, about 5 days. At this stage then, I'd like to draw your attention to the inside of the blastocyst, where there are cells called the inner cell mass, which I'll be abbreviating as ICM. Those are the cells that make the entire animal. And the outer cells give rise to the placenta and other supporting tissues. At this stage, the embryo implants into the wall of the uterus. This is when a pregnancy is really initiated. And now we'll see those blue inner cell mass cells form a disc, and then as the cells continue to grow, they change their physical positions, their kind of geographical relationship to one another. And you'll see that represented here as this disc gets transformed into an embryo. Those lines represent sites where cells are migrating in and out. And here's an important stage when the three beginning layers of the embryo, the so-called germ layers, are formed, and I'll come back to that in a few minutes.
So this is that primitive streak in this migration.
As development proceeds, there's more growth and movement of cells. It'll begin to form a neural tube. Here it turns, and appendages start to bud out. You see the head forming and the eye. And then eventually we get a small embryo, and some months later, of course, this would be born as a young baby.
So I know that's kind of still kind of a little bit surface treatment of that whole development and implantation, but you know, it's pretty interesting, and I mean, there are a lot of, let me close this off. Okay, so does anyone have any other questions, or do you want to go back and see the movie again, or otherwise I'm going to move on? Oh, I see a question, yes.
Just so that we can know like what to remember, you want us to know like what the ectoderm, mesoderm, and then the other one, like where they lie in the body cell?
Yeah, I mean, I want you, you know what, if you download the notes, I just put my notes up online. So I'm just gonna give them my notes. You need to read this. If you read this, you'll be fine. If you understand everything here, you'll be fine. And what I have here is really just notes on the primary germ layers. I want you to understand that gastrula is approximately day 16, and from which all other more specialized tissues are later derived. The outermost layer, the ectoderm, the innermost, the endoderm, and between these two layers and developing slightly later lies the mesoderm. And at first, the three layers are more or less uniform. Soon, however, they begin to take on different characteristics. And then I give a further definition of gastrulation, is the process in which a gastrula develops from a blastula by the inward migrations of cells by a complex and coordinated series of cellular movements which occur at the end of the cleavage stage of development. And that's what he showed you, that picture of gastrulation. That very simplistically, that's all I really want you to understand, is remember how he drew the sea urchin, and you get the mouth and the anus. And isn't it nice how we go through the world mouth first? You know, it really helps us a lot. I would not like to have to go through the world the other way around. So, you know, we need to think about these things. You probably never thought about your body plan being, oh yeah, my body plan puts me mouth first through the world. And that's, you know, important. Something to really notice. Okay, and then the cells begin to differentiate into the three primary embryonic germ layers. Ectoderm and endoderm first, followed shortly thereafter by mesoderm, during which the cells which differentiate into endoderm migrate inward to form the primitive gut which is called archenteron, and which subsequent cells differentiate into mesoderm within the blastocele cavity. And so that was, kind of what you saw, that blastocele cavity, and then this. And the process of invagination, and involution is the process of the cells moving. What those cells moving causes is invagination. We have invagination. I mean, that's what the fist going through the balloon is. That's invagination. I mean, we have, that's a very, I want to say, a lot of our body parts, they've been formed that way. It's just this folding in, this, just like it sounds, just invagination. Amniotic cavity is the amniotic fluid-filled cavity surrounding the developing embryo and fetus within the uterus. I mean, that's all you need to know about that. The ectoderm, the outermost layer of the 3 primary germ layers of an embryo, which develop during the transition from blastocele to gastrula, and from which the epidermis and epidermal derivatives come from. So that's kind of what is important, and then the other 2 layers.
So you want us to know the notes from the embryology and as well as the PowerPoint from histology?
Yeah, it says embryology of tissues, terms and notes.
And the PowerPoint as well, right?
Really, the PowerPoint is just kind of a supplement, and it gives us something to talk about while we're in class here. I don't expect you guys to memorize the PowerPoints. They're just, to me, they're just the show that I'm giving you, the kind of visuals. But in terms of embryology, I think this is pretty simple. I'm not asking you to, learn about all the genes, and although it is pretty fascinating. Karen Crow does some incredible work in our department on evolution and development. Karen Crow, Carmen Domingo, they're kind of what we call the Evo Devo kind of queens. Evolutionary development, and we have some great courses over there. And so I'm just giving you the little taste of what this is, but enough to just so you'll understand when we talk about anatomy and these organs and their forming. And of course, the oogenesis and morphogenesis is just formation of organs and the change of organs. How do the organs change over time? Okay. Any other questions? Okay. Well, I didn't know. I need to give you guys an apology because I'm used to doing this in 45-minute lectures, and now I have I feel like we're getting 2 weeks out of a week, and I'm like, wow, we're already there. So part of me is like, oh, let's just go home. But I know, but we have a half an hour. So if it was 15 minutes, I'd let you go. Let's see, Lucy, that's not as soon as she sees me walk by the cookie jar, or we call it the woof jar, then it's like there is nothing else in her mind except the cookie. She's 14. She has cancer. I think I'm going to lose her this semester. She's not doing well at all. So she's a lovely, she's a wonderful dog. I just dearly love her, dearly. Anyway, so I can't find my skeleton. So you know what? Let's just spend a few minutes talking about. So did anyone have any questions on the lab report or anything like that you?
Did you want it handwritten or typed?
Oh, you mean the little assignment?
Well, not the assignment, but the...
Oh, lab reports in general. I prefer them typed because they're easier to read. So your lab instructor will let you know. Anyone have any questions on the skull lab? Some of you haven't had it, but most
Not
of
on
you
the lab,
have.
but on the Apple.
Oh, yeah,
Did you want that turned in on iLearn or?
I prefer it here because I lose track if you try to turn it in electronically. I'm afraid. I'd like it turned in. Yeah, so, actually, you know what, it's Friday, no, it's Thursday. It's Friday to me.
Friday for this class.
Right, for this class.